Excellent. Thanks so much, Dr. Carstens. It's my real pleasure to be here today, and I'll also just start by thanking the organising committee. I think this is just such a wonderful, thoughtful and innovative and accessible forum, so really it's my pleasure to be here. I'll just pull up my slides. Lisa, I hope you'll interrupt me if anything seems amiss. I'll also start today by acknowledging that I'm presenting from Turrbal land in Brisbane, Australia, and just recognise them as the ancestral owners of this land. Today I'll be talking about model misspecification in microbiome studies. If you have questions, I'm really looking forward to the Q&A after this session, and if you'd like to get in touch with me, you can do so by email or Twitter, or I'm now on Mastodon. So the story that I'm going to be telling today really begins with the advent of high-throughput sequencing, which, as many of you know, has revolutionized the way that we study biology and the way that we study medicine, essentially by enabling far cheaper and far faster generation of enormous quantities of genomic data, and that's in biodiversity studies, in immunology, in basic sciences, really across the board of biological sciences. So one of the things that distinguishes high-throughput sequencing from perhaps traditional ways that we've studied ecological communities is that it's an indirect way of obtaining information about the environment that we care about. And so let's think about how maybe someone would have done a study into the composition of the communities of frogs living in a river. Someone back in the 70s, for example, someone might have sat there with a clipboard and a hat taking note about how many frogs they saw and at what time, and how many. And a slightly different experimental procedure is happening when we go to study microbiomes using high-throughput sequencing. So after having collected and stored samples, potentially after having had to wait some time to accumulate enough to justify sequencing, we then have to lyse bacterial cells, breaking them open and pulling out their DNA. Depending on the type of sequencing that we're doing, we might be selecting for a particular amplicon, whether it's 16S or ITS, maybe 18S, depending on what type of what domain we're looking at. Potentially, we may not have enough DNA, so there may be an amplification step. Once we've got this sufficient quantity of DNA, we'll go on to sequencing. And after obtaining our raw sequencing data, perhaps in the form of FASTQ files, we'll be processing those sequences into quantitative information in a process typically referred to as bioinformatics. And so, as you're probably aware, at every one of these steps in the experimental process, you have to make decisions about how you're going to do this. So what extraction kit are you going to use, and what type of sequencing are you going to do, and what bioinformatics pipeline are you going to use to process your sequencing data into quantitative data. So it's worth thinking about how much do those decisions that you make impact the results that you get at the end of the day. So let's take a look in the literature and see if, for example, the temperature of the freezer and the duration of storage impacts the resulting microbiome profiles. And there's paper after paper showing choices made at this step do in fact matter. What about DNA extraction kit and how we choose to do lysis, whether we choose a bead beating step or not, for example? And there are tens of papers, potentially hundreds of papers, about how your choice of DNA extraction methodology does have a really significant impact on the profiles that you get at the end of the day. And then what about the decision to do 16S versus shotgun sequencing? And if we choose to do 16S, what region of the 16S gene do we look at? This also has a really big impact on the data that we get at the end of the day. And so I think that this puts us in this kind of difficult place if our goal is to be doing science and to be studying medicine, because you have to make choices in order to run an experiment, and you've probably made the choices that you believe are the best. However, your colleague down the hall is probably going to disagree with you about what's best and have made different choices. And so, while you can believe strongly in yours, someone else may not agree with you. And so some of the discussion about how do we reconcile these problems has come from the framework of, well, we all need to agree on a universal set of experimental protocols, and we should all carry out studies according to those same protocols. So I think this is challenging for two reasons. The first is that it's hard to keep protocols static because, for example, extraction kits depend on companies that make them and then they're changing their offerings. So it's hard to keep that consistency over time. And then furthermore, I think it's unrealistic to think that different types of samples are best amenable to the same types of process. So how we study human fecal material is probably going to differ from how we should be studying saltwater ocean environments, for example. But really, at the end of the day, the main problem with this paradigm is that when studies disagree on maybe the direction or the magnitude of an effect size, for example, it's really hard to either confirm or refute a scientific claim. So reconciling different pieces of information across different experiments, because those differences can always be attributable to differences in experimental protocol, it's really hard to figure out what's actually going on in many different complex microbial settings. And so the scope that I'm going to be focusing on today is to talk about a model for how we go from the microbial communities that we care about studying to the sequencing data that ends up on our desks at the end of the day. And so really this story is about calibration and about evaluating the accuracy of community composition profiles, and I guess how trustworthy our OTU tables, for example, are at the end of the day. And if they're not so trustworthy, what can we do to improve them? So that's what I'll be talking about today. So I'll try to keep the notation pretty light in this talk, but I'd love everyone to think about a OTU or taxon table. So we can be thinking about tables of samples by taxa or strains, and I'll think about Wij as denoting the number of times that we observe strain J in sample I. Nothing about the talk that I'm going to give today is predicated on talking about amplicon data or 16S data. And if you're a metagenomics person, you can equally well replace these count values with coverages from metagenomic sequencing, either through a reference-based or not approach. So what this table means, just to make this really clear, is that in this particular study, we've observed 19 copies of Lactobacillus crispatus in this first sample. We've observed 4 copies of Lactobacillus iners in this first sample, and we've observed 0 copies of Lactobacillus crispatus in this second sample. And so a really common assumption that underlies a lot of methods for the analysis of microbiome data is the following. It's that observed relative abundances are proportional to some community-level true relative abundances of those strains in that community. So I'll let Pij denote some relative abundance of strain J in sample I. And so we can think about this as the counts that we end up with, or the coverages that we end up with for a particular strain, as being the product of some true relative abundances and some sample-specific, maybe library size or sequencing depth, sample-specific term that I'll call CI. So some methods that rely on this assumption include DE-seq, include LEFT-seq, include corn-cob. It's really a very prevalent assumption. And so a natural question to ask is this a reasonable assumption? Is this a reasonable model to be basing a lot of our microbiome methods on? And so a great tool that we have for evaluating assumptions in microbiome studies are mock communities. So mock communities are artificially constructed communities of known composition. You know what's in a mock community because of their construction, and their construction can be validated using optical density measurements, for example, and other cell counting types of tools. And so a mock community data set might look as follows. So we might see in this first sample here that our community is comprised 100% of Streptococcus agalactiae. Our second sample is comprised 50-50 of Aptopia vaginae and Prevotella bivia. And then samples 3 and 4 are 1-to-1-to-1 mixtures of 3 other taxa. So if you've worked in microbiome science for a long time, it may or may not surprise you to learn that OTU table, that ASV table that I showed earlier is actually derived from the mock community data shown in this lower figure here. So I'll take a second to just let that sink in. And I'll then make 2 observations that I think are pretty important here. So the first is that we're observing strains, or observing ASVs in samples in which they should be absent. So for example, we observed 19 copies of Lactobacillus crispatus, despite the fact that it should be absent in sample 1. The second observation I want to make is that we're not observing counts of taxa in proportion to their abundance. So let's focus on sample 3, where we have 33% and 33% of Lactobacillus crispatus and Lactobacillus innes, and yet we're observing about 2.5 times more copies of Lactobacillus innes than Lactobacillus crispatus, so about 11,000 copies compared to about 4,800, despite there being equal copies by cell numbers of those two taxa. So those are important points. I'll just say them one more time. What we see and what we learn from this mock community data is that despite equal mixing fractions, we observe some strains many more times than others. And furthermore, despite strains supposedly being absent, we observe them in those samples. Okay, so what we're going to be talking about for the rest of this time is using this control data to propose and then validate a more sensible model for microbiome data, quantitative microbiome data. So I'm going to propose a model for where our counts and where our coverages come from that essentially decomposes those expected counts into 2 pieces. So firstly, we've got this piece of the counts contributed from what we think should be there. And then we've got this piece contributing things that sort of shouldn't be there. So we can think of that as contamination in a couple different forms. So I'll start by talking you through where this contribution from sample piece comes from in more detail. So I've pulled out a couple of the mock communities from the data set that I showed earlier. So I've got 5 different mock communities here. On the top panel, I've shown their true composition, their true relative abundance as they've been constructed artificially. And then in the lower panel, I've shown their observed relative abundances that came out after 16S sequencing. So what these 5 samples have in common is that they all contain the same 2 taxa, this green taxon and this blue taxon. So that's Lactobacillus innes and Streptococcus agalactiae. And if we look at them, we observe that despite the fact that we always have a 1-to-1 or equal mixing fractions of this green taxon and this blue taxon, we're consistently over-observing this green taxon relative to this blue taxon. So let's look at that sort of quantitatively. We see that across every one of those samples, we observe about 18 times more copies of Lactobacillus innes compared to Streptococcus agalactiae, and they're in 1-to-1 mixtures in all of these samples. So that's pretty impressive. I think that's a pretty consistent signal, especially compared to other signals we sometimes see in high-throughput sequencing. And so is that a specific feature to these 2 taxa? So let's take a look across a couple of different taxa, a couple of different pairs of taxa. So here I've shown our Lactobacillus innes and Streptococcus agalactiae, and the fact that we tend to observe about 18 times more Lactobacillus innes to Streptococcus agalactiae, regardless of what other species are in that mixture. And I'll now show you across a bunch of other different pairs. And we see that while we observe, we do not observe this 1-to-1 ratio of one taxon to the other, despite that's what is present in all of these mixtures. We observe a fairly consistent over-detection of one taxon to the other that's specific to the pairs of taxa that we're looking at. Okay, so Lactobacillus innes compared to Gardnerella vaginalis, we tend to observe about 30 times more copies of Lactobacillus innes compared to Gardnerella vaginalis. Okay, so what I'm going to argue is that this data provides pretty compelling evidence against this model where expected counts are proportional to our true relative abundances with the proportionality factor as being sample specific, having some sample-specific library size or sequencing depth term, and appears to be much stronger support for a model that accounts for which taxon we're considering. So essentially what we've got here is that in addition to the product of our true relative abundances and some sample-specific sequencing intensity, we've also got some taxon-specific detectability term that I'm going to think of as EJ, as being a pretty important player in the expected counts that we tend to observe. Okay, so let's just think through really quickly what the implications are for this model on observed relative abundances. So let's no longer think about the count scale. Let's go back to thinking about relative abundance scale. And what we have is that our true relative abundances are proportional, our observed relative abundances are proportional to our true relative abundances multiplied by taxon-specific efficiencies. We can kind of forget about the sample-specific term because we're now working on this relative abundance scale. So what are the implications of this for analyzing microbiome data? So let's think about sort of a noiseless, really clean experiment where here we're looking at maybe a treatment group compared to a control group, and we're trying to reduce, our goal with the treatment is to reduce the relative abundance of our pathogenic, say, orange taxon. And what we see here in our observed data is a decrease in the relative abundance of our orange taxon from 42% in the control sample to 24% in that treatment sample. However, a true system that could have generated this data actually shows the opposite, where in fact there was an increase in the relative abundance of our pathogen from 20% to 33% after administering our treatment. So how could this be happening? Here's the following relative detection efficiencies that could have generated this data. So the setup is essentially that we have one really easy to detect taxon, this green taxon. It's 18 times easier to detect than our blue taxon, which is in fact the values that we saw for Lactobacillus innis compared to Streptococcus agalactiae. And then our orange taxon is sort of medium easy to detect, 6 times easier to detect than our blue taxon. And so because the relative abundance of this green taxon is changing, as well as the relative abundances of our orange and blue taxa are changing, we see that the change from 5% to 33% in the relative abundance of our green taxon in treatment to control, combined with its extreme easy to detectness, sort of eats up the relative abundance pie that's available and sort of edges out this pathogenic orange taxon from showing us its true relative abundance, at least in the direction that we care about. And so this is really misleading us as a result of these detection efficiencies combined with changes in other relative abundances. And so really, I just want to emphasize that this model has real implications, I think. This model that we've validated in this mock community data has real implications for not only the magnitude of effect sizes that we observe in real data, but also their direction. And I think that is something I would love everyone to think about going forward. And so just to wrap up this piece in terms of contribution from sample, what we see is that, or what I've argued for, is that our contribution from sample depends on 3 things, not just our true relative abundances, which is what we care about, but also some sample-specific sequencing intensity term, as well as some taxon-specific detectability or efficiency term. I'll move pretty quickly through this piece of contribution from contaminant or spurious sources. Essentially, the model that we are thinking about is that there's some contamination profile as well as some contamination intensity. And then there's potentially multiple of these contamination sources. And then how much contamination we observe in them is also proportional to how deeply we're sequencing that sample. So if you have further questions about this, I'd love to chat about them in the Q&A. But the result at the end of the day is that we have this model with 2 pieces, contributions from sample, contribution from spurious sources. The reason why I put all of this matrix algebra here is really just to emphasize that this is an extremely flexible model that can handle technical replicates, it can handle dilution series, it can handle multiple batches, it can handle whatever hypothesis you have about where your different parts of your experiment that are contributing different pieces of contamination. So that's a really exciting piece. I think the other thing that I'm really pleased about with this model and something I'm really proud of is that the relative abundances that we can estimate in this sort of correction model, correcting for detection effects, allow us to estimate true relative abundances that are exactly equal to 0. So there's nothing going on under the hood regarding pseudo counts or assumptions that all taxa are present in all samples. We can estimate strains to be absent in some samples if the data suggests it. Okay, so if you'd like to know some more details about this model, I'd encourage you to check out this preprint. It's called Modeling Complex Measurement Error in Microbiome Experiments. Really all of the hard work and all of the fantastic ideas for this came from David Clausen, who was a PhD student who I was lucky enough to work with, who graduated early this year. In this paper, you'll also see a couple of data examples that I don't have time to go through today, but some of the ways in which we've shown that you can use this model in your work include in deciding between different experimental protocols. So suppose you're setting out to perform a really big sequencing experiment and you'd like to decide how do you want to go about doing your DNA extraction, for example. So this is a model that can help you frame that question and choose amongst different protocols. Another is to estimate specific detectability of different taxa. If you have, for example, mock community data, you can estimate which taxa are easiest to detect, and then you can do correction on samples in which you don't know the composition. And then, of course, if you're really interested in contamination, as I mentioned earlier, there's also an example of how you can use this model to remove contamination, especially in the presence of data structures like technical replicates, or something we're really advocating for is dilution series. And I know that our host and moderator, Lisa Carstens, also has some thoughts about this. So that paper is fairly focused on the modeling pieces, but maybe if you're looking for a more intuitive way of understanding this model, I'll point you somewhere else, and that's another preprint led by Mike McLaren, who's I've been really lucky to work with for a few years now. He's now based at MIT. He was formerly at NC State working with Ben Callahan. The paper is called Implications of Taxonomic Bias for Microbial Differential Abundance Analysis, and I would say it's a really accessible introduction to the idea of detectability, and this particular preprint talks about the impacts of taxon-specific detection effects on both relative as well as sort of absolute, like qPCR technologies. Another thing that's really, I think, a great contribution of this paper is that it characterizes the situations that give rise to the problem I spoke about earlier, where you have some taxa, or is it the case that detection effects can lead you in the wrong direction in terms of the magnitude of effect size, so leading you to believe that there's a decrease in relative abundance when in fact there's an increase, for example. And it's also really nice because there's quite, I think, specific and clear recommendations for how you might do your microbiome data analysis in the presence of some of these artifacts. So do check out this paper as well. So there's a lot of work that I think is ongoing and important, sort of in the vein of this model. I think one thing I'm really especially excited to work on in the next few years is guidance for you all regarding experimental design. And so how should you choose between positive and negative control data? Should you be doing mock communities, or how should you be allocating sequencing effort between mock communities and blanks? Or sort of along the same lines, should you be running technical replicates, or should you be running dilution series? How do you want to make your decisions in that vein? Another thing I'm really interested in is, well, it's never going to be feasible to make extremely diverse mock communities. So we're never going to have all taxa represented in mock communities, and thus we may not be able to estimate the detectability of all taxa. And so I'm curious to see how much genomic information that we have about taxa can allow us to predict their detectability in the absence of being able to culture them, for example. I'm also really excited about this analysis question. How do we go about comparing different samples, given that we have unequal detectability of taxa and then uncertainty in estimating this corrected relative abundances? So that work should hopefully be out in the next few months, and you can follow me on Twitter or Mastodon if you'd like to see that as soon as it comes out. And then I really think the holy grail of this field is calibration and meta-analysis. So how can we be improving power to detect true differences in microbial communities by pooling data across multiple experiments, across multiple populations, but in a way that corrects for the fact that they have almost certainly been making different choices in their experimental process. One thing I'll add is that in order for this to be feasible, in my view, we really do need to have sort of community buy-in. So folks need to be collecting and reporting, essentially, relevant control data in order to make that possible alongside their experiments. So a lot of folks collect mock community data but don't make it available alongside their sequencing samples, and that is something that impedes this sort of cross-study comparison. So I'll close with a quick summary before acknowledging support and collaborators. If you take nothing else away from this talk, I really want you to take away that observed relative abundances are biased estimates of true relative abundances. And I've talked about 16S data today, but the same is true with shotgun data. And I have some supplementary slides showing that if folks are interested. So there's a couple of implications that this has for the analysis of microbiome data. So for example, I talked through that a toy example where proportions changing can lead to studying proportions can lead us to conclusions in the wrong direction. So it seems like we have an increase in relative abundance. In fact, we have a decrease. Another thing that's really important to note is that these efficiencies vary across taxa and even across taxa that are relatively phylogenetically or taxonomically constrained. And so we saw an example earlier where we had 2 different lactobacillus species that differed by a factor of about 2.5 in their detectability. And so I think there are implications of this for aggregating, instead of looking at the strain level, choosing to aggregate to the genus level or to order or family levels or even phylum levels, because we're averaging across taxa that are differentially easy to detect. So I do have some skepticism about, for example, Bacteroides to Firmicutes ratios, which have been a common medical diagnostic. I think there is some hope here. There's some hope sort of on the analysis side, and then there's some hope here on the correction side. So it does seem like with the right control data, we can correct for these effects and obtain good estimates of true relative abundances. And of course, there are alternatives as well for us to analyze our data, for example, using ratio-based approaches, some of which I think are stronger than others, in a way that's robust to these sorts of effects. Happy to talk more about that in Q&A. This work has only been possible with some absolutely incredible collaborators and some absolutely incredible student collaborators that I've worked with over the years. So this modeling microbiome measurement error, modeling complex measurement error in microbiome experiments is joint work with David Clausen, who I mentioned earlier. In addition, Ben Callahan and Mike McLaren brought me into this a few years ago with this paper, Consistent and Correctable Bias in Metagenomic Sequencing Experiments. And I'll also highlight a paper by Ben and Mike, as well as collaborators Jacob Nearing and Karen Lloyd, that talks about implications of this model for microbial differential abundance analysis. I'm also very grateful to the National Institute for General Medical Sciences for funding this work. I didn't have anything else to add to the formal part of this talk today, but I'm really looking forward to this Q&A. So thanks so much for calling in.